Hello everyone. Today I will talk about the challenges in the approval of complex Otic and ophthalmic generic products from bioequivalence perspectives. My name is Chunsheng. I'm in Office of Bioequivalence, Division 3, within Office of Generic Drugs in CDER. I joined our division in 2014. The goal of today's talk is first to describe the agency's current thinking regarding in vitro bioequivalence determination for complex Otic and ophthalmic drug products, and second to discuss challenges with in vitro BE determination for these drug products. First, let's take a look at what complex Otic and ophthalmic drug products are. According to description in GDUFA 2 commitment letter, complex Otic and ophthalmic drug products include the dosage forms of suspensions, emulsions, ointment, and gels. These dosage forms are considered having complex route of drug delivery. Suspensions and emulsions are liquid dosage forms, and ointment and gels are semi-solid dosage forms. Next, we will see some examples. On the left of the slides are some examples of Otic suspensions, such as ciprofloxacin and hydrocortisone Otic suspension, ciprofloxacin Otic suspension, and ciprofloxacin and dexamethasone Otic suspension. On the right are some complex ophthalmic drug products, such as dexamethasone and tobramycin ophthalmic suspension, latanoprost ophthalmic emulsion, bacitracin ophthalmic ointment, and temolol-maleate ophthalmic gel forming solution. For complex Otic and ophthalmic drug products, in recent years, the agency has revised PSDs for quite a few drug products to include in vitro options for BE demonstration. In today's talk, we will first take a look at the general recommendations for using in vitro option to demonstrate BE, and then look at two cases to further discuss the challenges. Please note that we will primarily focus on Otic and ophthalmic suspensions and gels in today's talk. For other complex Otic and ophthalmic dosage forms, the general recommendations may also apply. However, depending on the API and drug products, additional or different studies may be recommended and may not be covered under today's talk. To qualify for the in vitro option under CFR 3/2024 the following three criteria should be met. First, the test and reference products should be qualitatively and quantitatively the same. Second, the comparative physicochemical characterization of the test and reference products should be acceptable. And third, the comparative in vitro drug release rates from the test and reference products should be acceptable. If any of these three criteria are not met, a comparative clinical endpoint study is needed. Please note that certain drug characteristics may impact recommendations for BE determination. For example, if the drug product contains an antibiotic component, a microbial cure rate study may be needed. Next, we will take a further look at the three criteria and how they are applied. First, formulation sameness. To meet this criterion, first, all inactive ingredients in the test product should be the same as those in the ILD product, and this is considered Q1 the same. Please note that here there are no exception excipients allowed. The reason is that this can mitigate the risk of potential differences in irritation or sensitization that may be caused by the test product using different inactive ingredients, and also mitigate the risk that the test product may have different interaction with the disease state. Second, the difference in the amount of inactive ingredients between the test and the ILD should be no more than 5%, and this is considered Q2 the same. A test product that does not meet the above criteria would not be considered Q1 and Q2 the same as the ILD. For a test product that is Q1, Q2 the same as the reference product, the next step is to compare their physicochemical characteristics. To ensure that microstructure of the test and reference products are comparable. At least 3 batches each of the test and RLDRS should be used for these studies. Depending on the APIs and drug product, a series of comparative studies may be recommended. From B perspectives, I will talk about unit dose content and particle size distribution studies. My colleague in OPQ will talk about other studies in details in the next talk. For unit dose content study, it should be conducted per unit dose for all APIs with a minimum of 10 units per batch for 3 batches each of the test and RLDRS. The unit dose content should be compared using population B approach. For particle size distribution study, similarly, a minimum of 10 datasets per batch, 3 batches each of the test and RLDRS should be used, and PBE on D50 and span or polydispersity index should be used for BE determination. The third criterion is to compare in vitro drug release between the test and RLDRS. This is to evaluate the effect of manufacturing differences. If the drug product contains antibiotic components, in vitro microbial cure rate study may also be needed. This is also to evaluate the effect of manufacturing differences on the antibiotic components. Next, we will see 2 cases. Case 1, an Otic suspension drug product. This product contains an antibiotic component, and the PSG recommends microbial cure rate studies in addition to formulation sameness, comparative physicochemical characterization studies, and IVRT studies. Deficiencies have been found in almost all the studies except formulation sameness. We will take a further look at these deficiencies for each study category. First, comparative physicochemical characterization studies. For these studies, pooled sampling strategy is not encouraged when single unit volume meets instrument requirement. When pooled samples are used, sufficient justifications should be provided. Depending on the drug product and how well the study is conducted, a possibly acceptable explanation is that the volume requirement by instrument exceeds the single unit volume. Sometimes we see that complete method validation for all studies were not submitted. Next, deficiencies related to IVRT study. We found the IVRT study can be very challenging. For BE demonstration, the IVRT method can be different from QC methods. For example, different USP apparatus can be used. In some cases, complete method development, optimization, and validation reports were not submitted. It is very important that important parameters be optimized and validated. For example, the selection of medium and USP apparatus, the volume for dissolution condition, flow rate selection if USP apparatus 4 is used, the sample stability, membrane selection and recovery, and on top of all, the discriminating ability of the method should be demonstrated. One possible method is use test product manufactured with APIs of different particle sizes. At least 12 units, 1 batch each of the test and RLDRS should be used. For microbial cure rate study, draft PSD for dexamethasone and tobramycin ophthalmic suspension has details regarding how the study shall be conducted. The organism selection should follow USP 51, antimicrobial effectiveness testing, and indications of the RLD labeling. We have seen deficiencies in method optimization and validation. For example, the dose selection, antibiotic carryover, colony counts of organisms on survival media. For pivotal study, we have seen inappropriate dose used. In the next slide, I will show a study result and explain why the dose is inappropriate. Sometimes raw data of colony counts were not submitted. Sometimes the statistical method is different from the PST recommendation. In this case, we recommend that detailed explanation of the statistical method along with the statistical report be submitted for further evaluation. Here is a result of a microbial curate study which used inappropriate dose. We can see that the study was conducted for up to 2 hours. At time 0, both the test and RLD-RS products were dosed into media with comparable concentrations of organisms. At the first sampling time point, 5 minutes, both the test and RLD-RS products showed a 100% reduction of the organisms. Although the test product has comparable cure rate as the RLD-RS product, BE conclusion cannot be made based on the result. The reason is that the cure rate is too fast, which can mask the potential significant difference between the test and RLD-RS products. We recommend that the dose be further optimized and the study be reconducted. A summary of Case 1. We recommend attention be paid to the following aspects. For physicochemical characterization study, in general, pooling sample strategy is not encouraged. For IVRT method optimization and validation, important factors such as apparatus selection, medium selection should be optimized and validated. It is very important that the discriminating ability of the method be demonstrated. For microbial curate study, dose selection should be optimized. Statistical analysis method should be in line with the current PST recommendation. Next, we will see Case 2 for an ophthalmic gel product. Case 2, an ophthalmic gel product. This product does not contain antibiotic component, therefore the PSG only recommends Q1, Q2 sameness, comparative physicochemical characterization study, and the comparative IVRT study. Deficiencies have been found to be related to particle size distribution study and IVRT study. We will take a closer look at the deficiencies respectively. For particle size distribution study, deficiencies have been found in the following aspects. First, for method development, we have seen sample preparation such as the range of dilution was not optimized. Sometimes the selection of instrument parameters was not explained. Please note that simply using manual instructions as the explanation is not adequate. Further justifications should be provided. For method validation, we have seen invalid validation samples were used. For example, for instrument performance validation, sometimes the test product was used. However, at the time of the study, the quality of the test product was unknown. Therefore, we recommend using the reference product as the validation sample for instrument performance validation. For BE determination, PBE approach based on D50 and SPAN, with data from a minimum of 10 units per batch from 3 different batches of each test and the reference products should be used. For IVRT study, we have identified deficiencies related to method development, optimization, and validation, which is similar to what we have observed for the Otic suspension drug product. For example, physiologically relevant medium should be used when possible. Dissolution and analytical conditions should be optimized and validated. The discriminating ability of the method should be demonstrated. In the next slide, I will show an example. For pivotal IVRT study, sometimes we see that the time points used for F2 calculations are inappropriate, and at least 12 units of one batch of freshly manufactured test and one batch of unexpired RLD products should be used. and complete release profile by the last sampling time point should be reached. Here is the slide showing two IVRT methods having different discriminating ability. On the right side is the method that is considered lack of discriminating ability. We can see that with this method, both the test and the reference products reached complete release within 3 minutes. This is considered too rapid, as it may mask some potential significant difference between the test and the reference products. On the left side is the method that is considered acceptable. We can see that with this method, both the test and the reference products released the APIs gradually. And there are 3 time points before complete release, allowing for F2 calculations. This method is considered having discriminating ability. So a summary of case 2 for ophthalmic gel product is that we have seen deficiencies in the PSD study and IVRT study. For the PSD study, attention should be made for sample dilution, optimization, and valid samples should be used during method validation, and a sufficient number of replicates should be used to collect sufficient data for PBE analysis. For the IVRT study, again, method discriminating ability is very important and should be demonstrated. And by the last sampling time point, complete drug release should be reached, and the time point used for F2 calculation should be explained. Finally, take-home message. Currently, for complex otic and ophthalmic suspension and gels, the recommended in vitro option reflects the agency's current thinking for BE establishment. There are challenges in PSD, IVRT, and microbial curate studies. For example, for PSD, method validation, sample preparation. For IVRT, method discriminating ability. For microbial curate study, dose selection, statistical method for BE determination. By paying attention to these aspects, we hope some deficiencies can be avoided and review cycle can be reduced. Next, let's take 2 challenge questions. Question number 1. Currently, the prerequisite for using in vitro options for BE demonstration is A. The test formulation is exactly the same as the RLD. B. The test product does not contain an inactive ingredient that is not contained in the RLD. C. The test formulation is Q1 and Q2 the same as the RLD. The answer is C. Question number 2. Currently, for particle size distribution study, the agency recommends how many datasets each of the test and the reference products for the PBE analysis. A. A minimum of 10 datasets from each batch for 3 batches each of test and RLD-RS. B. A minimum of 5 datasets from each batch for 3 batches each of test and RLD-RS. The answer is A. Congratulations to all who answered correctly. Finally, I'd like to take this opportunity to thank the following people: April, Bing, Qi, Wendy, Darby, Mohab, Zhiyang, Li, Yun, and Yuan for their all kinds of help during data analysis and the preparation of the presentation. Thank you all so much for your attention, and please submit your questions, and we will take a look and have the QA session. Thank you.
Good afternoon. My name is Poonam Chopra, and in the second part of this session, I will be presenting on the challenges in the approval of complex Otic and ophthalmic generic drug products from quality perspective. The learning objective for this presentation is to highlight the role of comparative physicochemical characterization from quality perspective to support in vitro bioequivalence studies, and discuss some of the critical studies that should be performed during product development for these complex Otic suspension and ophthalmic gel drug products to ensure quality of the final drug product. This slide shows the definition of complex drug products, and bullet 3 provided the definition of products with complex route of delivery such as ophthalmic and otic dosage forms that are formulated as suspensions, emulsions, and gels. For more complex drug products where manufacturing conditions and processing steps, etc. could affect the properties of the final drug product, the bioavailability may be measured or demonstrated by several in vitro in vivo methods. Now, FDA may require in vivo or in vitro testing, or maybe both, to measure the bioavailability of specific drug products per CFR 320.24. So I'll start with the first case study for Otic suspensions. Now, Otic suspensions are generally used to treat external and middle ear infections such as recurrent otitis externa or otitis media in pediatric and adult population. Now, these ear drops generally work by stopping the growth of bacteria and hence reducing the ear swelling and discomfort. Now, Otic suspensions are required to be sterile and the API is suspended in the formulation. Now, it helps the API to stay longer at the site of administration and hence improve the bioavailability. However, this could pose a problem with the physical stability. For example, particles may grow in size over time, which could give problem in resuspension and hence impact on the homogeneity of the drops which patients are getting. So in recent years, approximately 40% of the published product-specific guidances have been there for complex products, and they recommend and provide public access to current thinking on bioequivalence approaches for the complex drug products. Now, this is a snapshot of content of product-specific guidance of ciprofloxacin and dexamethasone Otic suspension, and this is just used as an example. Now, the text shows the recommendation for in vitro option, and as mentioned earlier also, that the first requirement is that the reference and the test product should be Q1, Q2 to each other. The second bullet of this product-specific guidance shows that the number of tests that should be performed for comparative physicochemical characterization and to establish Q3 similarity between RLD and test product. The third part includes the comparative in vitro drug release testing, and then finally fourth bullet includes the comparative in vitro antimicrobial kill rate study of both RLD and the test formulation. So from quality perspective, I'm going to discuss the second part of this product-specific guidance, and in addition to that, I will also provide an overview of other quality tests which are very important in establishing the critical quality attributes of the drug product, ensuring the high quality, which in turn support in establishing the in vitro bioequivalence. So as discussed earlier, that Q3 sameness is determined by comparative physicochemical characterization of the test product and RLD. Now, if any of these criteria are not met during the characterization and comparative testing of RLD and the generic test product, then a clinical endpoint study is needed. Now, this slide shows that physicochemical characteristics needed for Q3 similarity includes tests such as crystalline habit of insoluble API present in the formulation, appearance, pH, specific gravity, osmolality and viscosity. Third test is redispersibility, and then soluble fraction of insoluble API in the final drug product, unit dose content of all APIs present in the formulation, and last is 3-tier particle size. Now, these physicochemical characteristics needed for qualifying in vitro option and are also relevant for the clinical attributes for the drug product. For example, information on the crystalline habit of the API provides information on the stability of the formulation during shelf life. Drug retention is affected by viscosity, and it determines the residence time of the formulation at the site of administration. pH is critical quality attribute as it may lead to tissue irritation and drug absorption through the different layers of the ear membrane. And osmolality, similar to RLD, is to minimize the irritation. Redispersibility is the time required to redisperse the suspension formulation. So these physicochemical characteristics are important in determining the retention, stability, drug release, and hence the clinical performance of these complex drug products. So we expect you to provide the comparative data for all these Q3 characterization parameters. Now, in this slide, I'm going to discuss the formulation development considerations for Otic suspensions. So we know that the Q3 similarity means that same components in same concentration are present in the same arrangement of matter. And since difference in arrangement of matter may arise due to difference in manufacturing processes, even if the formulation is Q1, Q2 with RLD. So during the formulation development, it is important to look at different components of the formulation and provide the formulation developmental study report accordingly. For example, API quality has a direct impact upon drug product quality and performance. Now look if API is present in the soluble form or if it's present in the insoluble form. Now, if your API is soluble in the drug product, then you should provide solubility profile as a function of pH and temperature. To mitigate the risk of crystallization or precipitation of API during the storage, information on the solubility for the drug product, appropriate solvent at the pH and temperature of process and storage and handling should be provided. If the API is insoluble in the formulation and stays suspended in the Otic suspension formulation, then the information pertaining to drug substance solid state stability is very critical. It's important to know if your drug substance exhibits polymorphism, and information should be provided on the polymorphic form of API used in the manufacturing of the drug product. And then same way, drug substance particle size stability is another critical quality attribute, and particle size is dependent on if the API is micronized or unmicronized, and you need to have a control established for the API particle size. There might be a need of size reduction step of your API during the drug product manufacturing process. So we expect that you include a 3-tier particle size test in your drug substance specifications. So here I have provided the example of crystalline habit of insoluble API, dexamethasone. Dexamethasone is known to exist in two crystal forms A and B with distinct X-ray patterns. In general, crystalline form and habits of drug substance may influence the formulation characteristics such as suspension syringeability, which is mostly mechanical. And the other important quality attribute is dissolution. The same drug substance with different crystalline form may have different intrinsic dissolution rate. Therefore, the recommendation made on comparative crystalline habit of dexamethasone is to ensure that dexamethasone used in the generic product is the same as the one used in the RLD. And it's expected that a comparative data should be provided from the multiple lots of RLD. So this slide gives a list of common studies that should be performed during the formulation developmental studies. In the interest of time, I'm not going to discuss all of these studies in details. The main point of this slide is that there are many studies which should be performed for formulation development, and some of them are for process development. For example, we know from Q1, Q2 perspective that which excipients are going into the final drug product, as RLD package insert includes this information. However, it does not provide information on the excipient grades and properties. So it's very important, as the difference in the grade of the same excipient may impact on the quality and performance of the drug product. For example, different grade of a viscosity modifier excipient may impact on the viscosity of the drug product. So it's very important to do your due diligence about the different grades of the excipients and demonstrate that difference in excipient grade and excipient properties will not impact the product quality and performance. Bottom line is that these studies are very helpful in demonstrating thorough understanding of your drug product and also the risks associated with your formulation and how these risks are mitigated. In this slide, I have again provided the tests for physicochemical characteristics for establishing Q3 sameness of the test product with RLD. And I'm mainly going to focus on the red bullet for dose uniformity or unit dose content test, where we most commonly see the problem and we issue deficiencies during our assessment. And after that, I'm going to cover a little bit additional quality tests such as assay impurities and so on and so forth. So during manufacturing, as well as in the final drug product, homogeneity of the Otic suspension is very, very important. So during the manufacturing process, we expect that you demonstrate the homogeneity of your drug product in the tank from top, middle, and bottom of the tank. For example, in the in-process specifications, you should include this test and provide the results. And also during the filling into the container, it's very important to look into dose homogeneity, which means that drug product is homogeneous in the top, middle, and bottom of the container. So in a traditional suspension product, upon sitting, the suspended particles will start to settle, and in some cases the particles may start to cake at the bottom of the bottles, and it becomes difficult to resuspend. So to ensure consistent dosing, the bottle must be shaken to redistribute the API. And generally, the container closures should be shaken for 20 to 30 seconds, mimicking the actual patient use conditions, and then analyze the samples from the top, middle, and bottom of the container closure system. So unit dose content test basically shows that container closure is capable of delivering the correct drop size to deliver the accurate unit dose content in comparison to RLD. And we recommend you doing it on 3 exhibit batches upfront, and they should meet the assay acceptance criteria. It should be done on 3 batches through the shelf life of the drug product to demonstrate that API in the container is homogeneous and the dose that a patient is getting through the product shelf life is uniform. So this comparative physicochemical characterization or in vitro BE study is also very helpful in establishing clinically relevant quality attributes for drug product release and stability product specification to ensure the batch-to-batch consistency through post-approval and throughout life cycle of the drug product. Now this is where the bioequivalence connects to the product life cycle. Typical product quality tests for release and stability include assay impurities, container content, particulate matter, container fill volume, and so on and so forth. So the slide gives the list of product quality tests during the release and stability testing of your final drug product. And these tests are the basic expectations you should provide in the drug product release and stability specifications. The tests in green font color are from the product specific guidance, but please note that the list of quality tests are the critical quality attributes of Otic suspension drug product. And overall, the acceptance criteria for all these tests in general should be based on the results of comparative testing of multiple lots of RLD. And RLD samples should be kept at the long-term conditions, and they should not be taken from the accelerated storage conditions. So with that, I will shift gears here and will take the second case study of ophthalmic gels. Ophthalmic gels are sterile semisolids for application into the eye in the space between eyelid and eyeball. API may be present in the solubilized form or maybe in the suspended form. As far as quality tests for ophthalmic products, USP 771 covers all the quality tests for ophthalmic drug products, including ophthalmic gels. So overall, ophthalmic drug products must meet all the relevant regulatory requirements and applicable quality standard prior to approval of the product. So this is again the snapshot of content of product specific guidance of loteprednol etabonate ophthalmic gel, and this is just used as an example. The text shows the recommendation for in vitro option, and as mentioned earlier, also that the first requirement is that the reference and test product should be Q1, Q2 to each other. And the second part shows the number of tests that should be performed for comparative physicochemical characterization and as a part of the establishing Q3 similarity between RLD and the test product. And the third part includes the comparative in vitro drug release testing of loteprednol etabonate from the test and RLD formulation. So same as before for Otic suspensions, Q3 sameness is determined by comparative physicochemical characterization of the test product and RLD. This slide shows the physicochemical characteristics needed for Q3 similarity for ophthalmic gel drug products, and it includes crystalline habit of insoluble APIs present in the formulation, appearance, pH, specific gravity, osmolality and viscosity, soluble fraction of insoluble APIs in the final drug product, rheological properties of the drug product, and 3-tier particle size. So these physicochemical characteristics are important in determining the retention of the drug product in the ocular region and stability of the drug product and providing information on the drug release and hence the clinical performance of the complex drug products. And we expect you to provide the comparative data for all these Q3 characterization parameters. And these physicochemical characteristics are very similar to the physicochemical characterization tests discussed for Otic suspension, as I mentioned before, except the test for rheological properties of the gels. As the ophthalmic gel drug products are developed, a lot of studies are performed to understand the product thoroughly, to understand the risk of product quality and hence the risk to patients. So it's important to document all the supporting studies in your ANDA and provide the data and address all the risks for the drug product quality are adequately mitigated. Now, in terms of formulation development and the process development studies for ophthalmic gels, the information is very similar to what is discussed for Otic suspension complex drug products. In the product-specific guidance also, the physicochemical characterization study is very similar to as that of Otic suspension, except the test of rheological properties. And also there are additional tests from quality perspective, which I will quickly give an overview after discussing the rheological properties of ophthalmic gel formulations. So gel is an aqueous continuous phase formulation containing a gelling agent which provides the semi-solid characteristics resulting in a solid material at rest and a fluid under shear stress. So what happens in some cases, the particles may start to cake at the bottom of the bottle and become difficult to resuspend. So in the gel formulation, when the product is at rest, that is, there is no shear, the vehicle has solid-like properties and the particles cannot settle. So if the vehicle remained as a solid, it could not be dispensed with a dropper bottle. The viscosity rapidly decreases when shear is applied and allows the material to flow easily through the dropper tip, and it shows the rapid return of the solid state after the shear has been removed. Now this rapid conversion from a solid to liquid and liquid to solid allows the formulation to be easily dispensed and inhibits the API from settling. For example, the loteprednol etabonate gel is a thixotropic gel which converts to a fluid form suspension upon application of shear stress, and upon removal of shear it converts back to the gel form rapidly. So when used by the patient, the drug product is expected to easily flow from the container closure system and when force is applied, but it should return to a viscosity sufficient to remain on the eye for a period of time after application. Also, patient acceptability and therapeutic effectiveness of the drug product will vary depending on how the flow properties or the spreadability of the drug product changes with the blinking of the eyelid. Now, these characteristics can be assessed through rheological tests, including a determination of viscoelasticity, flow viscosity in response to shear, and temperature modulus relationships. So it is expected that you provide a detailed rheological behavior of your test formulation, demonstrating the flow properties of your formulation, or in other words, thixotropy, and how different stress rates impact on the viscosity of the formulation. And again, a comparative rheological profile and yield stress data is expected between the test product and the RLD, demonstrating similar rheological characteristics of both the formulations. So next is the test for uniformity in container. It's the degree of uniformity of the amount of active drug substance or API within the container. And this test should conform USP3. The uniformity of dosage is demonstrated by assay of top, middle, and bottom samples obtained from a tube or container cut open to withdraw respective samples for drug assay. So it's very important that you properly document the sample preparation methods and also the test method validations in your application. And the assay should be performed from the beginning, middle, and end of the container. And the test should be controlled in the drug product release and stability specifications. So uniformity of semi-solid formulation in gel form is a product performance test. And the next is unit dose content data. So this test is a quantitative test, and it should be there to ensure that consistent dosing of the drug product from the same container should be delivered as per the label claim. So this is the list of product quality tests during the release and stability testing of your final ophthalmic gel drug product. These tests are the basic expectations you should provide in the drug product release and stability specifications. A detailed talk was given a couple of years back discussing the quality attributes for ophthalmic drug products in one of the SBIA's presentations. So I would recommend you to refer to that for detailed information on the ophthalmic drug product release and stability specification tests. In conclusion, it's very important to deeply understand your drug product formulation development and establish the critical quality attributes accordingly to ensure the high quality for Otic suspension and ophthalmic gel drug products. Comparative physicochemical characterization testing of generic and RLD product is necessary for development and quality control of the test product, and it can also support in vitro bioequivalence approach for the test product. So it's important to provide adequate supporting data in your end applications to ensure smooth and timely assessment of your applications. So in the end, there is a challenge question. Currently, the comparative physicochemical characterization of RLD and complex drug products is performed to: option A is to demonstrate sameness of test with reference product in support of in vitro BE determination. B, justify critical quality attributes for drug product release and stability. C, assess that the test formulation is Q1, Q2 to RLD. And D is both A and B. And the right answer is D, which is both A and B. With that, I would like to thank my supervisor, Patricia Onimba, my office management, Bing Kai, Pahala Semumara, Susan Rosenkrantz, Andre Raw, my colleague Asif Rasheed, and SBIA organizers. Thank you all for your time and attention, and I will see you in panel discussion.
Good afternoon, everyone. My name is Mingliang Tan. I'm a staff fellow in the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD. It is my great pleasure to give a talk in this session. My talk will be focusing on using PBPK modeling approach to support generic ophthalmic drug product development and regulatory decision making. The objective of my talk is to recognize approaches to demonstrate bioequivalence or BE for ophthalmic generics, to explain challenges we are facing in developing and assessing ophthalmic generics, to recognize the utility of using physiologically based pharmacokinetics or PBPK modeling approach to advance the development and approval of ophthalmic generics, and to narrow down the knowledge gaps in this area through research projects. Why do we care about ophthalmic generic products? Let's first take a look at the data from a recent publication by my colleagues, as shown in this table, which summarizes the numbers of reference products, or RLD, and approved generics on the US market in terms of different dosage forms. As you may see from the table, we are doing pretty good for the generic ophthalmic solution products, where more than 50% of the RLDs have approved generic products available. However, for the complex formulations such as suspensions, emulsions, and ointments, we still have a long way to go. In terms of the suspension and the ointment formulations, the numbers are 23% and 30%, but actually, more than 75% of these suspension and ointment generics were approved pre-Hatch-Waxman Act before 1984, which was before we have robust generic approval pathways. For ophthalmic drug products, the determination of bioequivalence can be challenging. FDA has recommended a variety of options that can be used to demonstrate bioequivalence for different ophthalmic drug products, including in vivo local PK studies, in vivo PD studies, comparative clinical endpoint or CCE studies, and in vitro B studies. Each B option has inherent benefits, risks, and limitations. Also, not all options are appropriate for proposed generics. For simple ophthalmic solutions, if the test and the RLD formulations are Q1, Q2 equivalent, B is self-evident, and a waiver of the in vivo study can be requested, while for non-Q1, Q2 formulations, an in vivo PK or comparative clinical endpoint study is needed. In terms of complex dosage forms such as suspensions, ointments, and emulsions, BE may be demonstrated through Q3 sameness for Q1, Q2 formulations. For non-Q1, Q2 formulations, the BE may be demonstrated through PK endpoint and/or clinical endpoint studies. There are multiple pathways of communicating with FDA during the generic ophthalmic development and submissions, such as product-specific guidance or PSG, pre-ANDA meetings, and controlled correspondence or CC. There are some unique challenges in developing and assessing ophthalmic generic products. Ophthalmic drugs are locally acting and may give rise to challenges in measuring detectable drug levels as such measurements in local eye tissues are often impractical, unethical, and cost prohibitive. In terms of local PK studies, the measurements are usually limited to aqueous humor for products which have a site of action in the anterior chamber and are indicated for cataract patients undergoing surgery. These very limited local PK data in humans are usually through sparse samples with high variability, which usually requires large sample populations. Comparative clinical endpoint BE studies can be used to assess local concentrations indirectly, but these studies have their own challenges, and the studies may also be confounded by the degree of patient disease severity and the challenges in measuring efficacy. Why PBPK modelings? PBPK models may integrate information about physiology, drug and product properties, existing in vitro and in vivo data to predict the local eye tissue PK and the PD of the drug of interest in humans, which allows for determining the clinical relevance of critical drug product attribute as they relate to a BE assessment. It may also be used to extrapolate to human models from preclinical species where relatively rich data may be available. The validated PBPK models may also be used to perform virtual BE simulations, which may be used as an alternative BE approach in the development of generic drugs in lieu of conducting a PD or clinical endpoint BE studies. In this regard, OGD has implemented some PBPK-related ophthalmic research by collaborating internally within FDA as well as externally through grants or contracts. Internally, FDA performs some internal research to meet the regulatory scientific needs. Externally, research grants and the contracts are announced regularly. These external research opportunities can be found through the link given here. In the following slides, I'm going to briefly highlight our recent effort on the PBPK-related ophthalmic research. First, let's take a look at the ophthalmic suspensions. One internal project was the application of our previously developed and verified ocular suspension PBPK model to study formulation effect on exposure with a focus on tear dynamics impact on elimination with 3 different particle size distributions, non-linearity of PK with 3 different strengths. We also studied the effect of viscosity on the dexamethasone distributions in ocular tissues and the plasma, suspension and solution formulation effect on exposure. This study demonstrated that solid particles presented in a suspension can not only help to achieve a higher ocular exposure, but also unfavorably raise systemic exposure. For ointment formulations, one research contract was awarded to Simulations Plus to simulate ocular ointment formulations where dexamethasone and fluoromethalone ointment rapid models were developed and verified. Parameter sensitivity analysis was performed to understand the impact of ointment formulation changes on ocular exposure. For ocular emulsion formulations, we have performed an internal project on the cyclosporine emulsion modeling. The purpose was to study the impact of emerging critical quality attributes on product performance. Two internally built models have been built to study the impact of surface tension, osmolarity, and viscosity on bioavailability and tear film breakup time. In the meantime, we also have an external research project with CFDRC to develop tear film models by including accurate eye anatomy and eye blink dynamics in the models to simulate drug delivery and transport. This model approach uses CoBio compartment model with CoBio Q3D capabilities in the modeling, and the 3D tear film modeling results are in line with our internal modeling work, as mentioned in the previous slide. Further model validation is underway through internal and external collaborations. Internally, OTR in the Office of Pharmaceutical Quality, or OPQ, has successfully manufactured 5 formulations with the desired globule sizes and viscosities. Externally, the absorption systems is applying the prepared formulations to rabbit to measure tear film and the tear film meniscus thickness in rabbits. The measurement will allow for model validation of previously developed cyclosporine emulsion rabbit model. In addition to the PK models we have just talked about, we also have some internal research effort on developing PBPK PD models to correlate the changes of brinzolamide ophthalmic suspension to PK and then PD effect. The preliminary model has been developed, but the model verification is pending on the data through internal and external collaborations. Internally, OTR has manufactured 6 suspension formulations with desired particle sizes and viscosities. Externally, the manufactured formulations have been shipped to absorption systems for animal study. The PK/PD measurements will use placebo, RLD, and the FDA-manufactured formulations with single dose and multiple dose. Another external project is to develop rapid PBPK/PD models and then extrapolate rapid models to human for ophthalmic products through a grant with CFDRC. So far, some PK and PD models have been implemented. Here is an example of terminal rapid models. In terms of the extrapolation of preclinical model to human, there is also another ongoing external research grant awarded to Simulations Plus. The main focus of this project is to develop and validate PBPK/PD modeling strategy to support translation from preclinical species to human for ophthalmic drug products by determining likely physiological differences between rabbit and human, mechanistically extrapolating rabbit models to human models, and then validating the extrapolated human models and ultimately improving the human model prediction. With all the effort, as we just briefly reviewed, there still exist some challenges in ophthalmic modeling, such as lack of direct eye tissue concentrations for model validation in human, lack of information on melanin binding in eye tissues, lack of information on metabolizing enzyme levels in eye tissues, and the lack of transporter consideration in eye models. Here are a couple of areas which may need further research. extrapolating models to human from preclinical species, as we may have relatively rich data available for animal model validations. Quantifying metabolizing enzyme and transporter protein levels in eye tissues, and then incorporating this data into eye models to improve model predictability. Developing PD model and validating PD models for IOP drugs, as there is a relatively rich IOP data available, and performing biorelevant and high-quality in vitro study data, which may be used for IV-IVE or IV-IVC modeling. Now, it's time for fun. Challenge questions. Question number 1. For topical ophthalmic drug products, which of the following dosage form has more than half of the branded name products which have generics approved by FDA? A. Suspension. B. Emulsion. C. Ointment. Or D. Solution. I will give you a couple of seconds to think about that. For those of you who choose D, you are absolutely correct. More than 50% of the branded name ophthalmic solution products have generics approved by FDA. Challenge question number 2. For topical ophthalmic generics, which of the following option is not often directly used to demonstrate BE? A. In vitro study. B. In vivo local eye tissue PK studies. C. In vivo systemic PK studies. Or D. Comparative clinical endpoint studies. I will give you a couple of seconds to think about that. For those of you who choose C, you are correct. In vivo systemic PK study is not often directly used to demonstrate BE for topical ophthalmic generics, as topical ophthalmic drugs produce very low to undetectable levels of drug in the systemic circulation. And even if drug can be measured systemically, often there is no direct link between systemic and local drug exposure levels. In summary, demonstrating bioequivalence for locally acting drug products such as ophthalmic products may be challenging, as measurement of drug concentrations at the site of action in humans may not be feasible. On the other hand, PBPK modeling approach can integrate human physiology, drug and drug product properties, existing in vitro and in vivo data together, which may be used to have a mechanistic understanding of drug absorption and distribution, to bridge the knowledge gaps in the generic ophthalmic drug development and assessment, and ultimately utilized in regulatory submissions. I would like to acknowledge my colleagues at ORS OGD and OTR OPCO, and also external grantees and contractors as listed here. Thank you all for your attention, and I may answer your questions during the panel discussion.
Thank you to all of our presenters for the great presentations. We'll now enter into the Q&A panel session comprising of our session speakers and also introducing Dr. Yan Wang, who's the acting team lead for the Complex Drug Substances and Formulation Team in the Division of Therapeutic Performance and ORS. In her role, Dr. Wang works with a group of interdisciplinary scientists developing PSGs, addressing controlled correspondences, pre-ANDA meeting requests, citizens petitions, and internal consults in the area of complex drug substances and complex formulations for various routes of administration and dosage forms. Also, we welcome Dr. Asif Rasheed, who's a senior chemist, pharmaceutical quality assessor in the Office of Lifecycle Drug Products within OPQ. In OPQ, he's been involved in the assessment of liquid-based dosage forms for oral, topical, parenteral, and ophthalmic routes, and has served as application technical lead for a number of applications. Finally, we welcome Dr. Kwok, who's a senior pharmaceutical quality assessor in the Division of Liquid-Based Drug Products. In this role, he acts as the application technical lead for integrated quality assessment of generic parenteral, ophthalmic, topical, oral, and inhalation solution drug products. For the past 7 years, he's been reviewing ANDA, Bio-IND, and pre-ANDA meeting packages involving complex drug products. Also, he served as an FDA liaison to the USP Packaging and Distribution Expert Committee for developing USP packaging chapters and standards, as a member for the development of FDA guidance for drug delivery performance of drug-device combination products. Looks like we have a few questions coming in right now. Our first round of questions will go to Dr. Zhao. And here's the first question. If an RLD product is withdrawn, not for safety reasons, how does the agency determine Q2 to RLD or RS if the ANDA sponsors do not have the RLD available to conduct testing for excipients levels?
Thank you for the question. This is a good question. So, in general, the agency guidance for industry referencing approved drug products in ANDA submission, which is published in October 2020, asks that the Q1, Q2 comparison is against the ILD product. However, in the situation that the ILD is withdrawn, not for safety reason or not available, in that case, the Q1, Q2 can be compared to the RS. I think for regulation purpose, the applicant should submit a request under 21 CFR 314.99B to waive this requirement for comparing to ILD product in this case, in this specific situation. Thank you.
Thank you for responding to that question. The next question for Dr. Zhao is the following. What is meant by complete drug release? 80 or 90%, etc.?
Thank you for the question. In general, we are looking at a mean drug release around 85% at the by the last sampling time point. In that case, we consider that the drug is released relatively completely. Thank you.
Thank you for responding to that question. The next question for Dr. Zhao is the following. How many time points are acceptable for the F2 calculation? And what is the reference guidance?
Thank you for the question. In general, at least 3 time points is needed for F2 calculation, and there is a guidance, the guidance for industry dissolution testing of immediate release solid oral dosage forms that can be used for reference for F2 calculation. Thank you.
Thank you for responding to that question. We have one more question for Dr. Zhao, and here it is. How many lots of test and reference should be tested when performing IVRT drug release when not specified in the PSG?
Thank you for the question. In general, for BE demonstration, we ask that for IVRT study, we ask for one test lot and one reference lot. For each lot, we ask for 12 units. This lot must be one of the three active batches that which the applicant characterize other physicochemical characterizations. It has to be one of the three batches. But for IVRT study, for BE demonstration, we ask for one batch test and one batch reference. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Chopra. And here's the first question. Is it necessary to monitor the drug substance PSD if it is micronized and fully dissolved in the drug product?
Thank you for the question. If your API is in the solubilized form, then generally there is no need to establish PSD. However, you have to demonstrate that your API will remain solubilized in the drug product over shelf life, and there won't be any precipitation. And in the case, if your API is not completely solubilized and is suspended in your drug product formulation, then you do need to establish 3-tier particle size as a control.
Thank you for responding to that question. The next question for Dr. Chopra is the following. For physical tests, such as redispersibility, how does one use quantitative measures to show equivalence?
It's a great question. Generally, when we measure or when we expect redispersibility from qualitative perspective, it should be, you should keep it in mind that the samples should be taken mimicking the actual patient use conditions. And when it comes to the quantitative measure, it is generally the test which is called unit dose uniformity or unit dose homogeneity test. And the basic expectation in this test is that the samples should be taken from the top, middle, and bottom of each container after shaking per label instructions. And these samples should be tested for assay using the assay acceptance criteria to ensure homogeneity of your active ingredient within the entire container throughout the product shelf life.
Thank you for responding to that question. We have another question for Dr. Chopra, and here it is. What aspects are to be explored under crystalline habit? For example, dexamethasone?
Thank you for another great question. Well, again, the general expectation is that you should provide comparative XRPD overlay spectrum data, and this should be provided for API, exhibit batches, USP reference standard, and RLD in comparison as a part of formulation development. And this comparative data, again, just ensures that if there are any changes in the polymorphic form of your drug substance during the drug product manufacturing processes. In addition to this, it is also expected that the polymorphic identification test should be included as a part of DS specification to ensure that the polymorphic form is consistent during the commercial manufacturing of the drug product.
Thank you for responding to that question. The next group of questions will go towards Dr. Tan. And here's the first question for Dr. Tan. Will the agency consider including PBPK models to establish BE for ophthalmics and PSGs in the future?
That's really great question. Thank you for the question. I think that's our goal. Hopefully one day we can do that. But even though currently
we still have some knowledge gap. But I think through the effort from all the stakeholders, including industry, academia, and the regulatory agency, I think one day we may reach that goal. Thank you.
Thank you for responding to that question. The next question for Dr. Tan. Does the model for cyclosporin emulsion take into account the changes in viscosity of the formulation after installation into the eye?
Very good question. Thank you for the question. The short answer for that question is yes. I believe for this cyclosporin emulsion models, some sensitivity analysis has been performed to study the effect of viscosity change on the exposure. You may find more detail from the publication, from that paper, through the link, the reference I gave in my slides. Thank you.
Thank you for responding to that question. The next question for Dr. Tan. From a mechanistic modeling standpoint, what dosage form do we have a better understanding among these complex ophthalmic formulations as mentioned in your presentation?
Good question. For the complex ophthalmic formulations, as I briefly mentioned in my presentation, there has been some recent modeling effort on the complex ophthalmic dosage forms such as suspensions, ointments, and emulsions. Among those dosage forms, there is a relatively good understanding of the formulation effect on ocular exposure for suspensions in the modeling. Specifically, some suspension formulation characteristics can be incorporated in to study the particle size, for example, particle size distribution, particle size, and the suspension viscosity on the exposure of local eye tissue. Once the models are validated, this formulation attribute can be directly related to the ocular exposure, which may be useful in establishing bioequivalence among suspension formulations. Thanks.
Thank you for responding to that question. We have one last question for Dr. Tan in this round. Has FDA received any regulatory submissions where ophthalmic PBPK modeling is a component in the submission package from generic companies?
The short answer is yes. We have started to see an increasing interest in, especially in pre-ANDA stage, such as in pre-ANDA development meeting packages submitted to the agency. This interest may include the ANDA development plan, such as alternative approaches for establishing bioequivalences, where PBPK modeling serves as one of the important elements, as in the totality of evidence to demonstrate the bioequivalence between RLDs and the proposed generics. Thanks.
Thank you for responding to that question. The next group of questions will be directed to Dr. Wang, and here's the first question. Regarding Q2 sameness, does each inactive ingredient need to be individually assayed?
Thank you for the question. So when we conduct the Q1, Q2 assessment, the formulations are evaluated based on the inactive ingredients and corresponding amounts present in the final drug product formulation on an individual basis. So yes, you will have to assay the individual inactive ingredients as appropriate. In addition, I'd like to take this opportunity to make 2 additional comments. First, if an ingredients are used as processing aids and applicants wish to include such information in the composition table, then those ingredients that are removed during manufacturing process should be clearly listed as such in the proposed formulation table to allow clear understanding. And second, sometimes we see applicants include additional information on formulation quality attributes such as pH or other characteristics in composition table or in Q1, Q2 control correspondences. Please note that those information are considered as outside the scope of Q1, Q2 assessment. The acceptability of such information is an assessment issue which will be determined during ANDA assessment post-filing. Thank you.
Thank you for responding to that question. We'll give Dr. Wang just a couple of seconds to adjust her mic while we pull up this next question for her. And here is the question. It was noted during the presentation that IVRT method for BE demonstration can be different from the QC method. Does the QC method also need to show discriminative ability?
Thank you for that question, and I hope my mic works better this time. So for most dosage forms, except when a dosage form is designed to have rapid dissolution, then discriminatory ability is always part of the method validation, regardless of the purpose. For bioequivalence or for quality control purpose, if IVRT is proposed as part of it, then discriminatory ability should be part of the validation, except if the method is only intended to show rapid dissolution of the dosage form. Thank you.
Thank you for responding to that question. We're going to move on to Dr. Rasheed for the next question. And here's the question. It's a long one, so it'll take just a moment to ask it. For unit dose uniformity homogeneity tests and Otic suspensions, it was noted that the shaking should be done per labeling instructions. However, certain RLD products simply note shake well. In the absence of any specific instructions on the label, what does the agency recommend? Can sample preparation prior to analysis use an extended period of shaking to ensure complete redispersion of the insoluble API?
Thank you very much. That's really a very great question, very good question. How do you demonstrate uniformity and homogeneity of dose when the label does not provide any specific instruction other than just to state that shake well? So I think we need to think of the purpose of this test. Like, why do we want for this to be performed? And the purpose is to ensure that when the drug goes into the hands of a patient and the way it is being used by the patient, so dose after dose, the dose that the patient is administering is consistent and homogeneous. So, and of course, it should be consistent and homogeneous as stated in the product label. So in order to ensure that, the test should be performed mimicking the actual patient use condition. And you can imagine that, in real life, a patient, we don't expect a patient to use any extended shaking or use of any machine or mechanical stirrer. So you can simulate in your labs how a patient would use the drug in a real life scenario. So we, anticipate that a patient should not be able to shake a bottle more than perhaps a minute or 30 seconds or 40 seconds. And this is how this test needs to be performed. So I hope this answers the question. Thank you.
Thank you for responding to that question. The next question is directed to Dr. Kwok. And here's the question. For Otic suspensions, stability in two orientations is recommended. Can an applicant provide stability in upright and horizontal orientation, taking into account the fact that the exposure area for the suspension product is more in a horizontal bottle in comparison with an inverted bottle?
Thank you for the question. This is a good question. So generally speaking, the orientations upright and horizontal are considered acceptable for stability testing of Otic suspension. However, having said that, in your ANDA submission, you should always provide justification or rationale for the orientations that you selected for your stability testing. Thank you.
Thank you for responding to that question. We have just a few minutes left, and we're going to go back to Dr. Rasheed to try to get in one more question. And here's the final question. The physicochemical tests recommended for in vitro BE for the ophthalmic gel isopropanol etabinate are quite different from those of the ophthalmic ointment for isopropanol etabinate. Could you explain why it is so?
Thank you. Thanks for the question. So the test for demonstration of Q3 similarity, which includes the comparative physicochemical tests, so they are recommended based on the characteristics of the specific drug product formulation or the RLD formulation, right? So and those tests which are recommended in each of these specific product specific guidances, for example, for loteprednol etabinate gel versus the ointment. So they are recommended keeping in mind what would be meaningful in demonstrating comparability of the test product with the reference product. So for that matter, these recommendations for Q3 characterization for in vitro physicochemical testings are different. And I hope this answers your question. Thank you.
Thank you for responding to that question. We're going to go back to Dr. Wang for a final question. And here's the final question. For ophthalmic gel IVRT study, FDA is expecting early time point data, less than 1 minute. Can we follow SUPAC SS to establish BE?
Thank you for that question. I think another way to understand your question is you are asking if the IVRT method for ophthalmic gel or ophthalmic products in general is required to be biorelevant, therefore looking at a very short time period for sampling time. So as Dr. Zhao has mentioned in her presentation briefly, currently the IVRT is part of the in vitro approach, which was developed based on totality of evidence. So therefore, in vivo relevance or biopredictability is always desired, but is not required, as long as the method is properly validated to show sensitivity, robustness, and discriminatory ability. Then with regard to the SUPAC SS guidance, that is not a guidance specifically developed for ophthalmic semi-products. So for ophthalmic gel in particular, I think one has to consider the ophthalmic gel in terms of drug release mechanism and general behavior is more similar to a suspension product versus a true semi-solid gel or ointment kind. So I think that would dictate how much you could maybe borrow from that guidance to help you to establish BE. Thank you.
Thank you for responding to that question. And that's all the time we have for questions in our final Q&A panel. We'd like to thank all of our presenters and panelists today for answering